Hello, and welcome back to Human Evolution. Today I want to talk more in detail about chromosomes. So chromosomes are something we talk about when we talk about mitosis and meiosis, and we talk about this idea that DNA, these lengths of DNA that we have in eukaryotes, will coil up into these packages that can be easily distributed as the cell is dividing, whether it's going to be undergoing mitosis, asexual reproduction, or meiosis, the production of gametes, right? So let's talk a little bit more about what the chromosomes actually are, how they're structured, what's important about them, what do we use our understanding of chromosomes for. So what is a chromosome? It is simply a continuous strand of DNA and the proteins that it winds around. That's it, right? Some RNA as well, but mostly it's this kind of framework and then the DNA coiled around it. Okay, so when you talk about your chromosome, you start with your DNA, right? And then that DNA is going to coil around a group of proteins called histones, and here's a little histone complex, right? And then those are going to coil up like a phone cord or something like that, and then those are going to coil up again, and then we're going to coil up into the final chromosome structure that we're so familiar with. So when we look at the chromosome, there are three parts that you need to know. There's the telomere, which are the ends, and these ends are actually really important because as DNA replicates, because of the way it replicates, there's always that RNA primer that's there. We don't really talk about the RNA primer, but in order for the DNA polymerase to know where to start, a little RNA tag is put onto the DNA. Well, over time, at the ends of the chromosomes, at the ends of the DNA, right, you have these little RNA primer sections that sort of just get cut off every time it's replicated, and so you lose little pieces of the ends. Eventually, when you lose too many of these pieces at the ends, you end up cutting into genes. So most of the ends of all these chromosomes don't really have anything interesting there as sort of a buffer, and this is part of what causes people to age. As you start cutting into these genes after you replicate and replicate and replicate and lose little bits and little bits and little bits, eventually some of the stuff you start losing is important. And I'm hoping that we'll be talking more about that. We may or may not, but telomere shortening is one of the things that causes aging, right? Now, we've also got our centromere, not to be confused with centrioles, right? The centromere, which were where the spindle assembly forms during mitosis and meiosis. But centromeres are the center of the chromosome pair, and this is where those spindle fibers attach and will pull these two chromosomes apart, right? And then you have a term called the nucleosome, and the nucleosome is the complex of the DNA and the histone proteins. So here are the telomeres on either end, and here is the centromere. Now we're also going to talk about regions of the chromosomes. You have the heterochromatin and you have the euchromatin. And the euchromatin is not as tightly coiled as the heterochromatin, and that's because there are readable genes in that euchromatin. Okay, so heterochromatin, tightly coiled, can't be read. Euchromatin, less tightly coiled, can be read. All right, so genes. Genes are regions of DNA that code for proteins, and so they do stuff, right? Now, there are other parts of the DNA. You've got regulatory DNA, and that is parts of the DNA that regulate when genes are read and proteins are produced, and so you can upregulate, which means to produce more. You can downregulate, right? And so some of your DNA is still really important, but it doesn't code for proteins. Genes, though, are the things that are the sections that code for proteins, and we'll talk a lot about genes, obviously, and we have talked about genes in the past. But you get genes in the regions of the chromosomes, and they occur in these kind of like bands. So you get what's called the banding pattern on the genes. This is another way that we use to sort chromosomes, right? To categorize them. So how big they are, where the centromere is, and what the banding pattern is like, the bands of euchromatin where the genes are and the heterochromatin where the genes are not, what that pattern looks like. So those are the big three things that we use to sort chromosomes. So, homologous chromosomes will have the same genes, maybe not the same alleles, and an allele is a different version of a gene. They may not have the same alleles, but they will have the same gene banding pattern. So, homologous chromosomes, same gene banding pattern, okay? And here's just another like, oh, look, here are genes for tall, right? Here's a gene for red, here's a gene for brown, right? For hair color, whatever. But you see, so these would be the alleles, and then hair color would be the gene. Okay, so how many chromosomes do humans have? Well, you should know the answer to this immediately. We have 46 chromosomes, but that means we have 23 pairs. The answer to this question is 46 because there are 46 of them, but there are 23 pairs. So here's one of the things we do with chromosomes. We organize them so we can make comparisons between species. And when we organize them, we put them into a karyotype. This organization here is a karyotype. So when you look at this picture, you can tell a lot about this individual, right? Right up front. And I would ask you to look at it and say, what's the thing that you notice right up front about this set of genes? This one belongs to a biological female, right? XX. Very obvious. So we know right up front what the biological sex is. Now, this one belongs to a male because you've got the XY. But chromosome sets 1 through 22 will be the same in terms of their banding patterns and their sizes for all humans, right? Because those are the autosomal chromosomes. And then the XY, XX pairs will be different for males versus females, okay? So when we arrange them, we're going to match them with their homolog. Remember, you get one from mom, one from dad, same genes. We're going to organize them from largest to smallest. We're going to look at the centromere position, and then within that largest to smallest, we're going to organize them whether the centromere is, I'm going to switch to the next slide really quickly, metacentric in the middle, submetacentric, kind of in the middle of the top, right? And then acrocentric, which means you just have tiny, tiny little P arms. And then telocentric, which means there are actually no P arms. So P is the short arm, Q is the long arm. Okay, so this is that organization. Second part of it. And then the third part of it is we match the chromosomes based on their banding patterns because that's showing us the genes. Okay, so homolog chromosomes have the same size, shape, and banding pattern. One from mom, one from dad. And here's an example of the banding pattern. So here's the centromere in this chromosome. Here's the banding pattern, right? You've got a band here and then 3 bands here. And these are the heterochromatin because they're dark. All right. That was the slide I just showed you about centromere position. And then here is a karyotype of a normal male. Right? Here's the X, it's a lot longer, here's the Y. And the Y is shorter simply because it mostly deals with just the proteins that turn on the developmental stuff for males, right? And so there's actually not a lot of genes on the Y chromosome because you really couldn't have anything very important on there in terms of human function because females don't have one, right? Now you'll notice that the chromosomes are kind of all, you know, bunched up and bent. That doesn't mean anything bad. Remember, they're kind of like yarn, right? So here we have 2 normal female karyotypes, 46 chromosomes, female. Okay, that's what this means. Karyotype, 46 chromosomes. The sex chromosomes are XX. Okay, now let's look at this one. This one is not normal. So the other thing that we do, we don't just compare different species. We look and see, are there any chromosomal anomalies or abnormalities in the human chromosome that might indicate some sort of genetic disease, right? And so in this case, here is the problem. We have a male, but the problem is that we have 3 chromosomes at 21, right? The 21st chromosome. This is called trisomy. Anytime you have 3 of one, it's called trisomy. And this, so here is 47 chromosomes. It's a male. We have 1 extra 21st chromosome. Okay, so that's how you read this. And what that means is that we have an individual who has Down syndrome, right? So trisomy 21 is Down syndrome. All right, that is it for this one. Short. I hope you enjoyed that, and I will see you in class. Make sure to ask any questions that you have. All right, have a good day. Thanks for listening.